can start. Okay. Right, yeah. All right. Good. Okay. Good morning, everyone. So, were all of you able to do the first collab? Can you just raise your hand who did, who were able? Okay. The ones who were not able to, did you not try or did you get an error? Please tell, because because if you're not clear about this one, then the rest won't be clear at all, right? So all of you were able to do. What happened? Okay, that is so here, right? The first thing is that you would need to copy the Allahabad workshop data, and what would happen if you clicked is it would go. Okay, I will. Okay, so this is what I'm going to do. I'll just use my cell phone hotspot. It's okay. Yeah, so you should get something like this. And then there's a couple of ways to download. One is you just click here and then download. It will download the data. It is about 536 megabytes. Okay. So you download the data, and then the data will look something like, so this is all the course material that I have. Where is the data? Yeah. Yeah, so this is the data that looks like, right? So this is what you get. It is 561.9 megabytes. Then I extracted this, and these are the directories, okay? All of you clear so far? Yeah, okay. And then what I did was um, I went to my directory and then created a directory called Allahabad workshop data, and then I uploaded it here. Okay, so once the data is uploaded, again, like this is showing how to do. Once the data is uploaded, then I'm basically running these things to link. It's because of my internet connection, right? So then it will ask you for your Gmail address that you had created. And I agree with this. Uh, right, then you hit continue to allow access. And then you hit continue again. Okay, so once this is done, so basically this will link these programs to your Google Drive, right? So the drive is mounted. Then we want to install these packages, right? So these are several Python packages which have been like used in neuroimaging, right? So Nibel, NumPy is a mathematical package. Matplot is people who are familiar with MATLAB. So then you run through this. You should not get any error. And then, right, so you are done. So you're linking the drives. So then does anyone know Linux or Unix? So in Unix or Linux, the command to list the number of files is called ls, right? So it shows that there are 4 directories: data already preprocessed, data preprocessed, data raw, and template, right? So that is a Linux command. I can again try to change and then see what you will get, right? If I change, this is another way to display, right? And so again, all of you should be able to get this. And if I want to further see what is inside, let's say the data raw directory, I just click this, right? It shows that there are 2 subjects, subject 1, MSCO1, subject MSCO2, right? So those are the 2 directories. Right, right, right. So these are different ways in which you can look at the data, right? So again, if you know the Linux command, you can also use this one, which will tell you for that there are 4 directories. Further, in each directory, what are the files or what are the directories that are present, right? Okay, so here it tells that the raw data has 2 subjects, MSCO1 and MSCO2, right? and if I want to find out what is inside MSCO1, I can click and it will tell there are two directories, anat and func, right? Again, if I wanted to know what is inside, I can always do something like this, and it will tell you that, okay, there is two directories, anat and func. In anat, there is this directory, this file, and in anat, in functional, there are all these files. Okay. Again, if you have any questions, please speak out, because if it's not clear now, the next lecture, you will be left behind, right? Okay. Okay, so pretty much, right? This is showing what is in each of these directories. And then it tells us what are these files, and then it tells you information about the anatomical and functional files. And in this case, the functional files, there is task, and then there is resting, right? Is this clear to everyone? Yeah. Okay. No problem, right? No. Okay. So then maybe just I will take 5 minutes to try something in the second assignment, which is D1B, right? Again, I'll share it with you later. But I want to like just test what you had learned yesterday. So let me just try. Again, it's just accessing the same data set. And there is, for some of them, like this one, are pretty fast. It's because I'm using my computer hotspot in US. Maybe not the best way to do. Okay. Okay. Okay, maybe after this one. Maybe after this one. Again, all of you should get like same kind of things. Again, this part is the same as we had done in the previous one. Okay, it took 1 minute. Now we have to install, just like in the previous one, we have to install this. Just remember, just remember that if you put an exclamation, many of you have used MATLAB, right? How many of you have used MATLAB? So in MATLAB, if you want to run something outside, use the exclamation point, right? So that is what it does here. So again, it installed the libraries, and then again, like what we had done in the previous, I downloaded the files, right, in the anat file, right? So this is the file name, right? And in here, what we can do is basically we are loading this file, and then we want to find out the properties of the file, right? So when we run this, we get all the information about this file, right? So um, as a so I'll tell you here. So the dimension is 224 by 256 by 256, right? So that is the dimension. And then, right, so it's called the data shape. Then the spatial resolution, like the pixel dimension, is 0.8, 0.8, 0.8, right? So that means the spatial dimension is this much. Can anyone tell me what was the field of view based on what we had covered yesterday? Remember, like the field of view, it is what we had covered yesterday, right? So I had said the field of view, to get the spatial resolution, you divide the field of view with matrix dimension, right? So from this, you know the matrix dimension and you know the spatial resolution. So what should be the field of view? Good. So you multiply the like 224 or 256 by 0.8, 256 by 0.8. So that is your field of view. Okay. Again, I don't want to cover too much now, but I will, like, the ones who haven't done one, please do it like before 2 o'clock, maybe during lunch. You can do it. Should not take much time, maybe 15 minutes. I think the biggest bottleneck is downloading the data and then upload and run through this, because in the afternoon we will go through this one and this, like I'll see if I can do another one where you can analyze the data, right? Okay, so I can. All right, so so today, for the first, like in the first 2 hours, I'm going to cover again about task task activation. And as I had mentioned yesterday, in task activation, what we are doing is we are looking for changes in signal intensity pertaining to the task, right? And what are the different ways in which one can present the task, right? What are the different ways? Anyone? Maybe, how about you? In the back? Block design is one. Very good. Block design, event related, and mixed. What is block design? Maybe her. Thank you. Yeah. When you show a stimulus. Okay, so there is at least two conditions, and each condition has the same duration, right? Good. Um, right. So um, is this a block design, or is it event related? Anyone? Madhav, right? Madhav. Do you think it's block design or event related? Make a guess. That's why. Like, hmm? No, block design. See, so what is the characteristics of event related? How much is it typically? The event is about somewhere between 500 millisecond to at most 2 seconds. Like very few event related study in fMRI is done, which is more than 2 seconds, I would say, right? So therefore, this is a block design.
For the webcasting.
Oh, okay. This one has a directory called slides, and in there it's lecture 2. Should I wait till you? Okay, okay, right. So again, these are when you are, I should say that, so the functional data sets are 3D plus time. That's okay. The raw SNR typically means for the anatomical, right? And this one, when I like had said MR, it's the functional MR signal. Right, that is the so that is then this is the SNR, right? So that is the net effect, right? And then what we have to do is that, like, depending on the signal that we collect, we can look at the variation. um, if you have to button press or do a task, how much variability would you see, right? Again, this is for task activation. Also, what we want to see is, um, if we were to scan a number of subjects, how much variation there is in different brain regions, right? Blue is the canonical, like the theoretical one. How much is the change in the frontal eye field, primary motor cortex, supplementary eye field, and the primary visual cortex, right? So for the same stimulus, these are the variation that we observe, right? So what could be the sources of variation? So these are 4 regions which have variations in the response. So what could be, like, based on what we had covered, what could be the reason that there is some difference in the different brain regions? Hmm? These have been normalized, right? So we identify that, okay, this is the primary motor region, this is the um, visual cortex, this is the frontal eye field. So these are regions. So the CSF should not get affected. Right. In a different way. Right. Okay. Hmm? Is um, okay. So I think you're giving very detailed explanation. I was asking for something very simple, right? Like, let's say we scan all of you, and these are different subject. Like, what could be? the reason that different regions of interest have slightly different response. Very good. So that is the main reason, right? So what you're saying is also quite right. But let's say among all of you, I was also scanned, right? My response may be different, and the difference would be not because, okay, it could be because I wasn't paying attention, but it could be because my heart rate and respiration rate and my blood vessels is not as elastic and fast as some of you young people. And therefore, what would get affected, right? There is neuronal firing. What is the other one? The hemodynamic response, right? So these changes suggest that when you are scanning different subject, people always assume, like in fMRI, that the difference is simply due to neuronal factors. That is not the case. It is neuronal as well as underlying hemodynamics, right? And so you can think of this as, let's say you were doing people, healthy subject with people who had stroke, right? Then in that case, if for some reason you are collecting data from the stroke affected region, the response would be much different, right? So what, okay, so now based on what we had discussed, what factors govern response? Very good. Stimulus properties. Very good. Say what she said. So, so the individual differences, the stimulus property, the level of attention or performance pertaining to the task, and then underlying factors like hemodynamics, right? So these are some of the main factors, right? Again, within each of them, there are further subtypes, right? If we look at hemodynamic response function, let's say you were to be scanned, right? And let's say last night all of you had partied very hard and had no sleep and had no breakfast and went to the scanner. Do you think that would be the same response versus yesterday? Like, like would like, so there are all these factors like food, satiety, like food, sleep, and many other responses. If there is like all these factors that affect the signal, but typically, like although there are changes, they are not big changes. Also, that is why it's important when you scan a subject, you know, you should ask them, right? If like the other example would be, let's say I was smoking, like let's say I was your age, but I smoked like 10, like 5 packs a day. What would my HRF be the same, right? So again, we don't, like when we collect data, like you should try to find all these things out, right? Because these are the main sources of variation in the fMRI signal. So now this is showing response to the same task across 2 different sessions, right? The same subject was scanned 2 times, and then like this is the 2 response, right? And so there is some variation, but there is going to be variation, right? Based on what I just mentioned, like they can have subtle differences. Also, as someone had mentioned, right, if you perform a task today, right, especially a complex task, right, and then the same task was shown tomorrow, then there is a habituation effect. So that is going to also play a role, right? Similarly, like movie watching and so on. Just imagine if you had seen a movie and the movie was shown to you, may or may not have the same effect. Okay. So there are many different artifacts that occur in fMRI, and so we had done a study where we scanned a large number of subjects between 5 to 8 years of age, again, healthy subject, and we scanned maybe 500 subjects. Make a guess how many subjects at the end I could use for the analysis. Uh, I how much? Okay, it's okay. If I was using 10%, then something is like my design is bad, right? I should have. So we could only use about 50%, 50 to 60%. Um, and like so, um, a lot of the subject, especially the kids, are the hardest because I guess they know a little bit. Uh, so now make a guess. What is the biggest artifact? Any head motion? Anyone else? Uh, you're right, right? So, and the easiest way to identify head motion is that if you see that you have signal around here on the edge, then it most likely is head motion, right? That is one way to judge. The other way is that if you're getting too much activity, then most likely it is head motion. Um, so there are different ways to look at the head motion, and believe it or not, the easiest way to identify head motion is to visually look at the data. And like um, one way is that I make a movie of the data, and if there is head motion, I can find where it is. That's one way. The other way is I look at the first image and the last image and toggle back and forth. That way also I can find if there is head motion, right? And like this is, again, this is a subject where these are the three translation and three rotation, right? X, like X, Y, Z, X, Y, Y, Z, and X, Z, right? So because it's a three-dimensional object, it can move in these different ways. And there are many algorithms that can be used to identify this, and we can fairly easily identify if there is head motion, right? So, um, right? So X, Y, Z, and then there is pitch, roll, and yaw, right? So where we can estimate this, right? And there are, um, many different ways to, people have tried to control for head motion, where they try like arm brace so that, for, again, I do a lot of, because I'm not a psychologist. I do very simple tasks. So like finger tapping and so on. So for finger tapping, many times when we would ask someone to tap, they would be lying in the scanner and suddenly they would do this, right? And that is going to cause head motion. So this is trying to put their hand like this and then ask them to just do the finger tapping. But as I said, if you do some kind of analysis and you're getting a lot of effect on the edge of the brain, then most likely it is due to head motion, right? So sometimes even when a subject tries their best not to move, there is going to be subtle motion. And some of the reason is that you have heart rate and respiration rate, right? Like respiration, right? So like your um, chest expands and contracts, that is going to move your brain by like zero point 0.1 to zero point 0.2 millimeters. And that is enough to cause subtle changes. So, yeah. And as I said, um, if you have a time course which looks like this, then be a little careful, right? So in head motion, the easiest way to find compared to task response is that in head motion, because it's a physical effect, the signal change is abrupt, right? While for fMRI signal increase, it's gradual, right? So um, that is like another way to identify head motion, right? So if you made a first order derivative of a signal, you would see a big spike for head motion, right? So this is um, right, a percent change. So, you know, again, if you are in doubt, then you should just not use the data. However, like I think that is where fMRI is so difficult, but also quite easy because you can choose whatever you want to do, right? Again, maybe for healthy kids you can throw, but let's say you wanted to study a population who had brain tumors, right? Or Alzheimer's, like finding each subject is so difficult that, you know, perhaps you would try to find if there is head motion while the person is in the scanner and repeat the scan, right? So in terms of head motion, maybe the final thing I would like to say is that in your analysis, if there is edge, then be very careful. Then most likely there is an effect. The the subject has moved. Maybe you can still identify, correct for it, and so on, but most likely the subject has moved. Okay, Dr. Bhumika, it's 11. Should I continue or okay? If the snacks is here, then they can take a break. So if any, okay, no problem. So if anyone wants to wants to become rich, if you can think of a way which would reduce head motion, I'm pretty sure you can become rich very easily, right? But the problem is that like how would you design so people have, maybe I should have put the slides. So people have used different ways, right? Some have used this cushions. Some people have also used like a mask from plaster of Paris, which would like till your neck, right? So you cannot move. But even then, if someone wants to move, they can still move. So that is one. The other thing is that there are studies now. So let's say you scanned a subject, like let's say you scanned five of your friends, um, and let's say A, B, C, D, E, right? A, B, C, D, E, yeah. So A has the most motion, B has second most, and so on, right? That's what you found. Let's say you scan them again the next time. What do you think will happen? Will it be the same? Will it be random? Everyone thinks it's random. No, the bad subjects, the bad subjects are always bad subjects, right? So if someone is, because someone did this study and like they found that there was this, uh, so they had scanned subject 2 different days. And what they found was there was like a correlation of zero point 0.8 on the motion from one day to the next, right? So the subject who had moved a lot in the first day had also moved a lot in the second day. But so some like so what they did was they found uh, like behavioral measures, like so people with high impulsivity typically have high head motion and so on. So it's like we should not be throwing away, right? So try to find ways to analyze. Um, so these are different ways in which, again, this is not exactly related to fMRI, but these are ways in which we should still scan as a quality check, right? So we should still scan like one of these phantoms to see that there's no effect due to like a RF coil not working or something not working properly. So um, like every week, most imaging centers do a QA test, right? So if you suspect something is off, what you should do is look at the QA images from week to week and see if those images look fine. Right? Um, right. So I think this is it. Yeah. So these are okay. Sorry, I had it, but I didn't. So these are different ways. One is cushion. The other one is a bite bar where you like bite to something which is attached to this one. Um, this is another one where right, and like this is you would have some distortion based on like susceptibility, and because you know the image how it should look like, this is a phantom and therefore should be a circle. So if you get an image like this or this, then you know what kind of image processing to do to get image like this, and then you can use the same kind of transformation on the anatomical image to get decent quality images, right? So like these are like, it's called B field corrections, right? So this is like the bias field estimation and correction. So from here you can find the B field and then do the appropriate correction. Similarly, you can use, if this is the functional, this is the anatomical. Again, with the functional, there is more distortion, so you can use the anatomical to do the correction. Right? And the again, this is showing, I think, um, as was mentioned, that in addition to hemodynamic differences, like different people have I think as she said, different people have different size of the head, and even the anatomical features are, the location is slightly different, and therefore we have to do different correction, or we normalize it to a standard brain template where we basically transform every single subject into this template called the MNI template, right? So again, this is like, so there is a template called MNI template, which is from like 200 or so subject, which has been like manually transformed and averaged, and then we look at these changes. So um, when we did the task, right, when we did the task, and you can do a t-test or correlation to find the activation. The other way is to do the Fourier transform, right? You can take this time series, do the FFT, and the fast Fourier transform would tell us that at this frequency there is a large signal, right? And you could use this information to map the brain, right? Is that clear to everyone? Okay. The other ways in which we can use is that um, once you identify the different activation region, we also use spatial smoothing, right? Temporal smoothing increases the signal to noise ratio. Similarly, spatial smoothing also increases signal to noise ratio. As I said, in temporal smoothing, when you smooth, you lose temporal information, right? So similarly, in spatial smoothing, when you lose fine specificity because you're averaging across different voxel, right? And so, depending on the spatial smoothing, you can get images which look like this or this, right? So you have to be a little careful, and you should also know the underlying anatomy well to understand which is what. So the other questions to ask is, I think as I had said before, are the independent variables appropriate, right? Um, you know, and then is the dependent variable appropriate? Like if they are not, then we should design good task, right? Assuming or making sure that both of these are appropriate, then we can do a hypothesis testing, right? And then we can proceed with the experiment. And then as I showed, right, you can have task A, task B, right, repeating. And then in one case, you have like carrot, mailbox, knife, tiger, sweater, and so on. And the other, you have these. Then you can design this to look at the responses, right? So this one, again, is task one. This is task two. So there are two variability, right? One is around here. The other one is compared to one condition. And so both play an important role, right? Um, so in this case, there is an increase, right? There is a decrease, like these are the baseline condition, control task, and task of interest, right? And as I had mentioned yesterday, when we are doing this, there is this assumption that during baseline, every region is the same, but that is not the case, right? Different regions have different intrinsic properties, right? Because they have different hemodynamic response function. The cerebral blood flow varies across different regions, also across different subjects. Similarly, CMRO2 and oxygen extraction fraction, right? And again, this is the default mode, and these are the areas associated with this. And again, this suggests that even when someone is not performing the task, these regions are active, right? When they perform this task, these regions become deactivated, right? Um, and these are like stimulus independent factors. And again, so while if you have a block design, you are basically stimulating at a single frequency, right? And if you're unlucky and that frequency happens to be at respiration or some other factors, then your detection won't be very good, right? So that is why um, perhaps the best kind of stimulus would be anyone. So periodic is easy to analyze because it's at one frequency, um, but it has some limitation that um, you can guess. Also, it is a specific frequency. So what would be an ideal stimulus? Hmm? Anyone? Hmm? Good. So you can jitter. So you can jitter, right? What is the other name for jitter? Very good. So what is, does anyone know what is the frequency? If you do the FFT of a random signal, what will you get? So if you have a sinusoidal signal, you do FFT, you will get one peak, right? The magnitude, frequency, and phase. If you have a random signal and you do the FFT, what will you get? So, so what is the definition of a random signal? Anything can happen anytime, you don't know, right? So therefore, in the frequency spectrum, what will you get? It will also be random, right? Because there is no, you don't know what frequency will occur. So in some way, I thought, is 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 there anyone who is an electrical engineer? Okay, doesn't matter. So when you have a random signal, um, this is perhaps the best design you can have of a stimulus, because um, like these events that occur are completely at random. The chance of happening, right, uh, by chance is very small, right? But the problem with designing a random stimulus is that, see, in a random stimulus, you have to have the off period random and the on period random. That is not a very good design, right? Because if you have on period of only 2 seconds, then the response won't go up, right? So, um, but so what people have done is try to make it like block slash random, um, to try to get better detection. Right? So the next thing, like once you have the activation map, the challenge is what threshold should we use, right? Yesterday I showed you a figure where you have the whole brain which is active, then you set a threshold, and based on the threshold, you say this is active, this is not. But where to set the threshold is a very difficult problem, right? Because you have type 1 and type 2 error. Does anyone know what false positives? What are false positive? Yeah, good. Right. So can you give an example? Can you give an example? Like it doesn't have to be MR related. Can you give an example of type 1 error? Right. Very good. Right. So that is type 1 error. Um, what would be the type 2 error? Yeah. Right. Right. So which one do you think is more dangerous? It depends on the application, right? Like, if, well, COVID is, well, no, no one talks about COVID anymore, right? So if one was to, whether one has COVID now or not, doesn't matter, right? But let's say if it was cancer, right, then maybe you would tolerate type 1 error more than type 2, right? Again, I think we are going to go through some examples later, right? Okay, no problem. Yeah, 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 no problem. Sometimes I feel I'm just covering something which is too basic.
But you have to learn it through, otherwise suddenly you appear like a jump.
Yeah.
It's okay.
Okay, I'll try to be.
Regarding physiological artifacts, I had a question.
Yeah.
So you're using stimuli, say for example, it's a reward context,
Yeah, yeah.
or emotional IAPS,
Right, right,
highly arousing or low arousing.
right,
So there you're manipulating arousal.
right, right.
It will lead to.
Yeah.
So
So,
how do you handle?
so do you have a biopack where
Yeah,
you
we
can
do.
measure the heart rate and
But
respiration?
we have not integrated it with.
Okay. So there are a couple of ways. One is you can you can retrospectively collect it from the existing data and see what is happening. So you can do that, but um, but maybe you should try to include. like if you can, because that is much more reliable.
Objective
Yeah,
measures.
yeah.
So
Some people,
otherwise, HRS will value.
right. Yeah. Some people also use skin conductance.
Ah,
Okay, so let's just start. So, as we discussed about type 1 and type 2 error, so next we are going to cover some of the basic analysis which is used for task activation, and these are called simple non-GLM approaches. And then in recent years, the general linear model is most commonly used, right? And there's another kind of analysis, across subject analysis, which is using fixed effect versus random effect analysis. Also, because we are using, like any given brain has so many voxels, even if you set a threshold at 0.01, just by chance there will be large number of voxels that will pass the threshold, right? And how do we correct for multiple comparison? And then what is the best way to display the data? So for simple hypothesis-driven analysis, the t-test is the simplest one, because like you are making an assumption between the two conditions, what the differences are. Again, it's like very simple and elegant, but the assumption we have made is that for the two conditions, everything remains the same, excepting the task that the subject performs. That may or may not be true for many cases. Also, as we discussed yesterday, in t-test, you look at the mean differences between two data sets or samples and then find differences, right? So the temporal information is not used, right? So like throwing away the temporal information is not a good idea in most of the cases. And the way to avoid it is using correlation, right? And finally, one can also use Fourier analysis, where you can use the frequency which is presented and identify, right? So using correlation approaches, you assume that when you present the task, your response is going to be a scaled version, maybe scaled and delayed version of the task, right? So the idea is if I tap my fingers for 20 seconds followed by 20 seconds of rest, my response is also going to be, 20 seconds of signal increase followed by 20 seconds of signal going back to baseline, right? And the amplitude of the response is like a fixed effect, right? So, so that is what is used for correlation analysis. So it assumes particular correlation template, as I mentioned. It cannot include task unrelated variability, right? If there is some variability unrelated to the task, you have X and Y, you cannot, account for that. Although there are other ways, right, to do it, but typically you cannot, okay? Also, it does not model interaction between events. If there is,
So can you please skip this PPT? Can you please skip
oh,
this PPT?
do you have a Type-C connector?
I have Type-C pen drive.
Yeah. Did anyone have any questions? It will just take a second. I have to. So these are some of the limitations of correlation analysis. Again, for the Fourier analysis, as I mentioned, you can switch back from time domain to frequency domain. And it is, the Fourier analysis is similar to correlation analysis. Right? And so, like, if you have a stimulus and you find the frequency analysis, then you can identify, the regions. Again, if these are two regions, the blue and the turquoise signal, if you did a correlation analysis between these two, what would you find? Can anyone make a guess? Like this two. Right, if you did a correlation between the two. So, so the correlation value is between -1 and +1, right? And so if one did a correlation between these two time series, what would you find? Is it -1? Right, between -1 and +1. But I think for this one, it would be, because they are out of phase, it will be 0, right? Because they are out of phase.
The same phase we will get the high.
Right, so if, again, if you have, in correlation analysis, if you get a correlation analysis, if you get a value of 1, it means that two signals, X and Y, are identical to each other, right? If you get a correlation of 0, they are out of phase, right? But if you get -1, they are the same signal, but they are inverted, right? So in correlation, you had.
What is meaning of this in terms of probability?
Right. So if you have two signals, right, and you are showing good correlation, that is good, right? Then your response is matching your task, and that is good. However, if your response and your task has no correlation, then it's not very good. However, if it's simply due to phase, then what does it mean physiologically? That you perform the task, but there was a slight delay, right? And therefore, it responded to the task. It was just that it was out of phase, and therefore it was 0, right? So to avoid such a situation, you can use the frequency domain analysis. By using frequency domain analysis, you could see that both right, these two have the same frequency, and this two have the same frequency. The only thing which is different is the phase, right? So this is a way to make sure that you're not losing something because of phase difference. I should say that people doing correlation analysis would point out that when you do the correlation analysis, it will also tell you at what phase would you get the maximum correlation value, right? And based on that, you could make an inference. Okay, so if you have two signal, right? So the challenge with correlation is that, let's say you have like an apple, like one apple, and then you scan the same apple which has been slightly shifted. If you do correlation, the correlation will be a little less because it has been shifted. However, if you have an apple and then an orange, the correlation will also be less. Just because it is less, it doesn't mean there is no effect, right? So one has to be careful about the phase information, right? And by doing frequency analysis, you're looking at like if it contains the same frequency. In task activation, what is more commonly used is called the general linear model, right? And in this case, what we assume is that, okay, or how we model is that this is the measured data set, so this is the fMRI. Actually, you don't have to write, maybe I can put the slides in a drive and then you can use it. So the fMRI response can be modeled as the design model, and this is the amplitude, and this is the noise, right? So in a very simple way, if we were to use correlation analysis, this one would be the stimulus, right? But with general linear model, we can have more complex models where we can model, right? So this is the data, this is the model, these are the model functions, amplitude, and this is the noise. And so these are the model parameters. It could be the images that you are shown, right? And then this could be the model parameters, and then you can have a design matrix, right? So each one, it could be, let's say you want to study visual response, it could be like, you could use something like this. But if you want to study faces versus places versus object, you could also design it so that here you could label which one is what, and then we can model it accordingly, right? So like if you have, like in this case, good, bad, good, like different conditions, and then you can model it and then analyze them. So at once you can find many different responses corresponding to different tasks, right? So these are from the design matrix which pertain to the different tasks, right? So if you want to, let's say between faces versus places would be one. If it's a simple visual input, it would be one, faces versus places, places versus faces, and so on. You could design these. Then here you could model heart rate signal, respiration signal, because these are, at least in this case, it could be nuisance parameters, right? But again, if you want to study where you want to say that, hey, if I show fear or disgusting picture, what would happen to my heart rate and respiration rate? Would it change, right? So then, if this is your heart rate and respiration, you could use them as your task regressors and identify, right? So this is a much more comprehensive way to study the task responses. And in here you can have multiple effects corresponding to the task. You could have multiple effects corresponding to nuisance regressor, which you think are noise sources. If you saw that the noise sources was 6 parameters, right? What would you guess are they? Right? So very good. Very good. So if there was 3, at least there would be 6, maybe more. So if it's only 6, my guess would be that it is translation, rotation, right? X, Y, Z, and then the 3. And so it can be used accordingly, right? And so in this case, you would have different hemodynamic functions, right? So there are many ways to describe the hemodynamic response function. One is a double gamma, and then the other ones are Gaussian and a single gamma function, which is basically a smoothing function which is used with your reference vector, right? Because again, if you're tapping, right, 10 seconds on, 10 seconds off, it would be a square. But when you tap your fingers and so on, the response is not going to be instantaneous, such that it will be square. So it will be convolved by the hemodynamics. And how to model that is by using one of these, right? And again, these are approximations, right? Because if we really want to understand the underlying hemodynamic function, we should do another run to find the hemodynamic function, right? So the optimal relation between regressor depends on our research question, right? Again, if someone was to study aging, right? So in that case, maybe the underlying hemodynamics is another regressor of interest, right? But if we were like all college student between 21 and 25, then maybe not, right? So depends on the question that you are asking. Right? Think of another example that, like, if, again, please forgive me for always bringing up finger tapping because, like, for me, that's like a very simple task, and it's a very robust task. But let's say instead of me, like there are different ways in which you can present a hemodynamic stimulus. One way is that, like you would say, tap, right, on the on the microphone, and you would hear, and then you would start tapping. But the other one would be maybe with stimulus design. You would have a visual stimulus, right? Which one do you think is better? Visual? Anyone? Visual, right? Because you have designed it using E prime, like it will be precise to the millisecond. While if I'm going to like scream like tap, then there may be variations, right? So let's say the stimulus, a visual stimulus was given, and then the person tapped their finger, right? Will there be a difference between the two responses, right? Perhaps because the first is visual stimulus, you see, and then you tap, right? And will the delay be 200 millisecond or 100 millisecond or 1 second? But there would be a delay, right? So in such cases, we can model this and have these values be presented such that we would still see a response. It would still have a good correlation, but because two are highly correlated. Again, this is the same idea we can mix around depending on the stimulus and create delay effects, right? Similarly, we can make regressors uncorrelated with each other through randomization, right? So again, in this case, if we make it quite like both of them are random, then the responses may look like this, and then when you do the correlation, then the value may be very low, right, as here. Right? So this is the Q regressor, this is the target regressor, and then again you can use different ways to some example would be averaging across subjects before a t-test, or taking all subjects' data and then doing an ANOVA. And with this, it allows inference to a single sample. So basically, the way it is done is, let's say you have 50 subjects who did a task. You take the time series across all the different subjects, average them, you get one average subject, and then you do the analysis, and then you get the combined statistical map, right? So this is a fixed effect. In random effect, you assume that the effect varies across the population, right? It accounts for inter-subject variance and analysis. It also allows inferences to population from which subjects are drawn, especially important for group comparison, right? So in random effect, you find the activation map corresponding to each subject, and then based on the number of subjects that you have, you get the same number of activation maps, and then you do your statistical comparison, right? So is there any difference between the fixed effect and random effects? In both the cases, you're averaging, right? So why would you use one versus the other? I can argue and say that, hey, if we had done finger tapping, this would be great because all the subjects are averaged, and if I wanted to see how the response is, I just go and click on the sensory motor cortex, and because of Okay, 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 she would use fixed one, right? Okay, why would you choose the fixed one? Anyone else who would use the fixed one?
Suppose population is of specific age group, then we can choose fixed. Or if population include all the age group people, we can look at.
Right. So let's say we use just all of you, right? Is it okay to use fixed effect? Yeah? What would be the limitation? Anyone? What is the um, what is the limitation of using fixed effect? Like fixed effect has this advantage that you are averaging everything into one subject, right? But what is the limitation of doing that?
In fixed effect, you're essentially comparing the averaged out between each participants, while in random effect, you're essentially seeing differences between all the participants in one condition versus all the participants in another condition. So it's more of the variability between the condition than it is between the participants across the conditions.
Uh, so just think that let's say we scanned and Sanchita moved her head a lot, right? And she was off by like 3 voxels, right? And then we average, what would we get? And let's say if it was just one, let's say 10% of the subject, like 3 of you, moved ahead too much, right? What would happen to the analysis? Would we see effect? We assume that the activation parameters may vary across subjects. Since subjects are randomly chosen, activation parameter may vary within group. Fixed effect model assumes that the parameters are constant across individual, right? So that is one. Again, this is just considering the same group, right? If we were to use, let's say, young versus old subject or healthy versus schizophrenia, then we would also have, we should also use random effects. So then is the issue of multiple comparison, right? So if, right, so this is p-value of 0.05. For these voxels, it is 1682 voxels. P-value of p 0.01 is 364 voxels, and p of 0.001, that is 32 voxels, right? So typically we use like p-value of 0.01. Again, this suggests that there will be several outliers, right? Or several false positives. So what we have to do is find ways to cluster the data, right? And like One easy way is to use spatial smoothing, right? And spatial smoothing, if there is a few voxels which are close to each other, then they are more significant than if they are randomly distributed, right? So with spatial smoothing, you can identify those to find there are 5 voxels and the 6th voxel is maybe 10 voxels away, then perhaps that voxel is just noise, right? So that is the basic idea with cluster analysis. There is ROI, like region of interest analysis, which is if I know I want to focus on sensory motor cortex, or if I want to focus on the default mode or the visual cortex, then I could just focus on those regions and then analyze the data, right? So with Bonferroni correction, this, as I said, is a very stringent measure because the way it works is that if I have 30,000 functional voxel, then it says my type 1 error should be 1 divided by 30,000, right? Which would be 0.0003. It's a very stringent condition. And so with this one, the challenge is that it introduces type 2 error, right? So you may lose voxels which are actually active. And again, this is not appropriate for correlated data. Like technically it's, it assumes that these data are randomly oriented, but the fMRI data is highly correlated data. So Bonferroni correction you can use, but you can, if you do some analysis which survives Bonferroni correction, then you have very good results, right? So the other approach is using Gaussian field theory, which was developed by Keith Worsley and colleague for multiple comparison. Analysis, or I did a like Keith Worsley's method and found this and asked you how many clusters are there, it would vary from subject to subject. And so in this case, what is done is we use clustering analysis as well as how many location that we are interested in, right? So in this case, we use like different cluster sizes, right? You can say that, okay, for something to be active, there should be 3 contiguous voxels which are active, right? And you would get a value. Or you can say that for it to be active, I want 5 voxels to be connected to each other, right? So you can define that way to find clusters, right? And as you define the number of voxels, you can find the cluster and set the threshold, right? So if you say, for me to identify something, 20 voxels should be contiguous, right? The chance that 20 voxels would be contiguous happening just by chance is very small, right? So then you can find the alpha value and report it in your paper, right? The other way is the false discovery rate. And the way this one works is that it controls the expected proportion of false positive value among suprathreshold values. So in this case, the number of voxels declared active, right? So in any data analysis, we only observe dA, dI, and V, right? So this is declared active, declared non-active. So this is one way you can identify, right? So as you set the threshold, these values would change. So this would be one way to find the optimal value. The other way in which we can do is using ROI-based analysis. Again, this is used but not very commonly used, because in this case, what you're doing is you're taking a region of interest and making it one value, right? So if you have, you can find a number of ROIs and then say that when you do this task, what happens, right? So if you define all this regions, right, and you say that between 2 different tasks, what happens to the amount of signal change, what happens to the mean value, and so on. So for you to identify one of this, that region would have significant differences. So in this case, you don't have to worry about multiple comparison, because it's like for each subject you would get one value, right? Any questions? Okay. Again, like voxel and RO. How do we visualize our data, right? Because we are collecting the data in 3D volume. Once you have the 3D volume, you can display in many ways, right? You can display along the orthogonal plane. You could also make them visible, like these are called the surface maps, right? Where you can display them on the surface of the brain and where they are, and you can have an idea of the depth and so on, right? This is another way where you can identify these and then display them on the activation. This is yet another way. This is mostly used by a software called SPM. They display their brain in this fashion, right? So this is axial, right? This is sagittal, and this one is coronal, right? Axial, coronal, and sagittal. And you can display them, right? And by clicking, it would show along different planes. By using surface, you can also display them in this fashion, right? This is by using the surface. This is the across the sagittal plane, and you can identify different regions of the visual cortex. This is something called the flat map, where you can display the cortex as a flat map and then identify the different brain regions. Right? So for the data analysis part, the basic take effect for multi like multiple comparison effect. Also, if we have to choose between fixed effect versus random effect, we should use random effects, right? I think I'll just stop here. It's 12:30 or 12.
We can have questions.
Yeah, okay. Anyone has questions? No question? Yeah.
In one slide you mentioned some related about phase that we put in code analysis.
Yeah.
So in that particular graph, I was not able to see that phase. Can you explain that? So you said that because phase shifted, so the correlation will be zero.
Okay, let me.
In 20 slides.
Okay. Okay, so in this case, there is the blue and the green, right? So from the frequency point of view, it's the same frequency, right? But they are out of phase, right? So when we do the correlation, it will not be positive, right? Because they are out of phase. And
Out of phase, from this graph, I'm not able to.
oh, so this is the response, right? So the blue is the response, the green is the response.
Sir, did you not mention that the correlation value will be 0 if there is a 90-degree phase?
Right.
This one seems more like 180.
180, right,
So
right.
maybe closer to -1?
-1, yeah. I think, yeah, you're right. So this one will be negative because when one is maximum, the other one is minimum, right? So how would, okay, so what would be the example? So the example maybe would be if I showed you a stimulus, in one condition, I would say when you see the stimulus, tap your finger, and I did a correlation between the visual signal and the motor signal, right? So they would be fairly correlated, right? There would be very little phase difference. In the second condition, I asked you that, okay, when you see the visual stimulus, do not tap. When you do not see the visual stimulus, then tap, right? So then they are out of phase, right? Yeah. Any other question? Okay, so maybe quickly, what is, like, can you tell some limitation of Bonferroni correction? Too stringent, very good. And? No, you said too stringent and? Can you speak a little louder? Oh, right. Yeah, so thank you. So too stringent and type 2 error, that you would miss actual voxels which are active. What else? What is the advantages of correlation analysis and disadvantages? Right.
correlation might not allow us to see where the peaks are, which Fourier transform would allow us to.
Okay, good. Any other ones? Any other advantages and disadvantages?
Interaction effect.
Right, so that's a disadvantage. What is, like, can you think of an advantage?
Simple analysis.
Very good. Yeah, it's very simple and intuitive, right? It's like you tap your finger, there is going to be a response, right? If you tap your fingers faster, the response will be, higher, right? But I think, as you said, the disadvantage is if the response is nonlinear, then you won't be able to model that, right? So the example that I was talking about, let's say finger tapping is okay, but let's say we were watching, or I made subjects watch like what to say, what is the word, like there are images like very bad images, like disturbing images, right? Like there's a database where you have, yeah, where you have photo of people. Hmm? Yeah, right. So if you have such images, maybe after a while, a person may get, may not have the same effect, right? Or in some other subject, the effect may be much more worse, right? And then the response would be higher. So as long as the response is a linear response, correlation is good. But if the response was nonlinear, or then you cannot use correlation analysis. Any other question? Yeah.
In the choice between the fixed and the random effect,
Yeah.
being also dependent upon the question that we are asking? I mean, the research question that we are having?
I guess if you want to see how the time series is, I guess maybe fixed effect would be good, right? If you wanted to see the images that we were talking about, if we want to see what happens, aggregate response to such stimulus, perhaps we can do like fixed, just to see the response. Maybe the other response would be, let's say your study was you inject cocaine to subjects and see the brain response, right? Again, you can only do it on people who are addicts. So in that case, the correlation analysis won't work, right? Because the response is going to be very different. It's not going to be linear, right? There will be a sudden peak and then it will fall down, right? And for those kind of thing, if you want to get a general response, perhaps a fixed effect would be okay. But having said that, now there are other methods like ICA or PCA which you can use to find common component, right? Because like these are averaging effects, right? And as I said, averaging is very good, but just for a second, let's say some like Bill Gates came into this room, like we all technically would be billionaires, right? But with what I'm trying to say is that with averaging, it's highly sensitive to outliers, right? And so for that, we should not, like we have to be very careful when we are using these methods. They're very simple, of course, but like we have to be careful. Okay. So please finish your assignment, and I will try to go through some of the other collabs, because I think once we do that, you will have a much better idea, because um, um, like we have spent quite a bit of time trying to organize the data and shown like different steps. And even after you finish this course, all you have to do is change the data, and I think it should still run. Yeah. Okay. See you at 2 o'clock. And maybe if you if you make good progress, the 2 subjects who were scanned, maybe, we can make it an assignment to analyze. That is one. The other thing was that these data, so I have also included like 100 subjects for the autism data. So I heard like some of you have done some work on autism, like from ITBHU, many students mentioned. So you can then check how does fMRI differences compare to structural ones. And then we have to have